Hello everyone, this video is about structure and function of major histocompatibility complex, and this study note is from Beach Pathshala site, MHRD, Government of India. So I would like to thank the development team for developing this study note. Thank you so much. Structure and function of major histocompatibility complex, MHC in detail. After studying this module, we shall be able to recognize the various haplotypes of MHC and their structures, differentiate between the class 1 MHC and class 2 MHC antigen presentation, learn the role of MHC in non-self recognition, ascertain about the genetic architecture of MHC, learn about MHC polymorphism and their basis on natural selection process, how epigenetics has a role in MHC polymorphism, know the role of MHC in autoimmunity and other disease conditions. Why MHC or HLA gene typing is essential for transplantation. Introduction. For the body to arraign an attack on any pathogen, it must be recognized as self or non-self, and it is accomplished by the set of cell surface receptors which not only distinguishes self from non-self but also plays a pivotal role in tissue transplantation and rejection, autoimmunity and susceptibility to infections. These cell surface receptors are encoded by the most diverse gene loci on vertebrate, known as major histocompatibility complex. So for the body to arraign an attack on any pathogen, it must be recognized as self or non-self, and it is accomplished by the set of cell surface receptors which not only distinguishes self from non-self but also plays a pivotal role in tissue transplantation and rejection, autoimmunity and susceptibility to infections. These cell surface receptors are encoded by the most diverse gene loci in vertebrates, known as major histocompatibility complex MHC class 1 and class 2 loci. In humans, MHC spans a considerable part of the DNA, about 0.1% of total genome, containing 200 coding loci. These are highly polymorphic genes that can be attributed to the natural selection over long periods of evolutionary time. The history behind MHC genes dates back to 1916, when Little and Taylor showed that tissue could be transplanted only in the same strain mice, followed by George D. Snell's observation in 1935 that several closely linked histocompatible genes are responsible for tissue rejection. He discovered that in tumor transplants on mice from their congenic cousins were immediately rejected but were successful in synergistic strains. The mystery was solved in 1975 by Zingernagel and Doherty, who found that T-cell responses were restricted to antigens and MHCs presented on the cell surface. The 1980s and 90s finally revealed the structure of MHC molecules and their encoded proteins. The Nobel Prize in Physiology or Medicine in 1980 was awarded jointly to Brugge Benacerraf, Jane Dosset and George D. Snell for their contribution in understanding that similar antigens are controlled by a dominant autosomal gene termed immune response genes located on the MHC loci of all the vertebrates. Other than any region of the genome, the MHC genes are the most associated with autoimmune and infectious diseases. These disease-associated variations in the MHC region are due to very subtle and minute changes in the gene loci. There is still more to learn about MHC, like what mechanism drives MHC polymorphism, which are naturally and positively selected variations having functional importance, which signaling pathways does the MHC genes follow at the molecular level. The MHC haplotypes. The MHC genes encode three classes of molecules. 1. MHC class 1. The MHC class 1 heavy genes are encoded by three genes, HLA-A, HLA-B and HLA-C, and present small peptides to CTLs, cytotoxic T lymphocytes, enabling each cell of the body to the immune system with possible chances of infection For every nucleated cell of the body present MHC1 molecule. Mostly these peptides are self-peptides, but in case of virus-infected cell or cancer cell or parasite-infected cell, the foreign peptides are presented, resulting in their elimination by CTLs. The CTLs are very unique cells with highly specific TCR T cell receptor that scan for MHC antigen complexes and CD8 plus molecules to stabilize them, help bind the cells to MHC antigens. The cells with foreign antigens help activate the CTLs, resulting in the clonal selection of these specific CTLs, thus eliminating other infected cells. But it is still very difficult to envisage that how MHC molecules achieve the specificity in immune recognition with such small pool of TCR. In fact, MHC molecules can bind to a varied range of peptides rather than specific peptides. Structure of MHC class 1. It is composed of alpha and beta polypeptide chains held together with non-covalent bonds with a short cytoplasmic tail. The alpha chain is the MHC encoded integral membrane glycoprotein of about 45,000 Dalton, while beta-2 microglobulin is non-MHC encoded extracellular 12 kilodalton peptide. The alpha chain is of 350 amino acid with 3 globular domains, alpha 1, alpha 2, alpha 3. Alpha 2 and alpha 3 both have intra-chain disulfide linkage. There also has a 26 hydrophobic amino acid transmembrane segment spinning the plasma membrane. The alpha 1 and alpha 2 forms the platform as the peptide binding unit, mainly comprising of beta sheets. The groove formed between them is blocked at both the ends and is enough to hold 8 to 10 amino acids, so it is imperative for the foreign residues to be processed and presented before they bind to MHC1. The beta-2 chain is non-polymorphic 90 amino acid peptide is encoded by chromosome 15 and interacting covalently with alpha 2. This part of MHC molecule bind to CTL. Class 1 MHC molecule is a glycoprotein with 1 or 2 N-linked oligosaccharides. Peptide binding groove of beta sheets alpha 1, alpha 2, alpha 3, beta 2M peptide hydrophobic structure cytoplasmic tail. This is the structure of MHC1 molecule. Then MHC class 2. So learn the structure. Alpha 2, alpha 1, alpha 3, beta 2M. Class 2 peptides are expressed on specialized cells called antigen-presenting cells, mainly the macrophages, dendritic cells, and beta B lymphocytes. MHC2 molecules are encoded by 3 polymorphic genes HLA-DR, HLA-DQ, and HLA-DP. DR, DQ, DP that bind to different peptides. The immunodominant epitopes are largely confined to the pathway dependent on lysosomal degradation and peptide binding of newly synthesized MHC2 molecules in the acidic endosomal compartment. The MHC2 molecules that are involved in the CD4 plus T cell response against intracellular pathogens may trigger immunity towards infection, thymic selection, autoimmunity, and tumor immunity. Structure of class 2 MHC. Like MHC1, the class 2 MHC molecules have an extracellular domain, a transmembrane domain, and an intracellular carboxy terminal tail. The extracellular domain is composed of 2 non-covalently associated domains, alpha 1, alpha 2, or beta 1, beta 2, 90-degree amino acid anchored to the cell membrane. The heavy chain alpha 1 and alpha 2 has a molecular weight of 30 to 34 kilodalton, and the light chain beta 2 and beta 2 has a molecular weight of about 26 to 29 kilodalton. Alpha 2, beta 1, and beta 2 domain contain disulfide linkages, while alpha 1, alpha 2, and beta 1 domains are glycosylated, but beta 2 domain is not. The transmembrane domain is comprised of 25 hydrophobic amino acids and the cytoplasmic tail of 10 to 15 hydrophilic amino acids. Peptide antigen binds in a groove formed from a pair of alpha helices on a floor of antiparallel beta strands involving alpha 1 and beta 1 segments respectively. The antigenic peptide binding domain can hold 13 to 18 alpha amino acid residues, and it is iso with both the ends of the peptide binding groove. Peptide binding cleft beta 1, beta 2, alpha 1, alpha 2, transmembrane structure, cytoplasmic tail. MHC class 3. These molecules are mainly comprised of serum protease and complement enzymes and do not have any role in antigen presentation. The complement component includes C2, serine protease, C4a and C4b, pro-proteins of the multi-chain forms, and factor B, component of alternate complement pathway. They also include the TNF alpha and TNF beta as well as two heat shock proteins. Structure of peptide bonding cleft. MHC1 and class 2 molecules fold into highly similar conformations featuring a peptide binding groove to present T cell epitopes. Peptide binding grooves of MHC1 molecules are composed of 2 alpha helices and 8 beta strands formed by one heavy chain, while MHC2 uses 2 domains from different chains to construct the peptide binding groove. Peptides bind to MHC molecules through primary and secondary anchor residues protruding into the pockets and the peptide binding grooves. Peptide preferences are dependent on the amino acid polymorphisms comprising the anchor pockets, which are related to the various alleles of MHC. The conformations of peptides presented by MHC1 molecules are length dependent. The modified groups of post-translationally modified peptides have important roles in the peptide MHC interaction. So here we could see peptide binding domain alpha 1, alpha 2, class 2, alpha 1, beta 1. Peptide binding cleft closed at both ends. Next one is open. So peptide binding domain and cleft of MHC class 1 and class 2 source, class 2 complex. Peptide MHC interaction. Peptide binding to MHC class 1 and class 2 molecules require classic anchor peptides at the N and C terminus of the peptide. These residues may be termed as anchor residues. These anchor residues or peptides bind to the pocket of the binding cleft of the MHC molecule. Class 1 MHC anchor resides are mainly comprised of hydrophobic amino acids. At C terminus, the anchor residues are at the 9th amino acid position, mainly comprising leucine and isoleucine, while the anchor at the N terminus is mostly present at the 2nd, 3rd alpha amino acid position. N-terminal anchor residue in H2KD is tyrosine, H2DD is glycine. The mid portion arches away from the floor of the MHC complex to make a contact with the TCR. Peptides that bind to class 2 MHC have 7 to 8 amino acid that provides maximum contact. It has hydrophobic residues at N terminus, C terminus as well as 3 amino acid in the middle, thereby providing a constant elevation from the floor of the binding cleft. Peptide in MHC class 1 binding groove, amino acid residue, peptide backbone binding groove. Peptide in MHC class 2 binding groove is like this. So peptide binding grooves of class MHC class 1 and class 2 proteins. Peptide binding in an MHC class 1 groove is usually 8 to 10 amino acid long. The residues at both the N terminus and C terminus end of the peptide are anchored in the groove. Peptides binding in an MHC class 2 grooves are usually 13 to 18 amino acid long. The residues bind at both the ends as well as at the middle. Class MHC class 1 and MHC class 2 antigen presentation. Both MHC1 and MHC2 present peptides to the CD8 plus and CD4 plus T cells. The sources of these peptides may differ. The MHC1 are presented with intracellular peptides and the exogenous peptides are presented by MHC2. Recently, it was noted that the cross presentation also occurs where the exogenous peptides are presented by MHC1 and endogenous by MHC2, evident by their overlapping characteristics. Both classes have similar three-dimensional structure. They originate from the common parent gene by gene duplication and a similar function in antigen presentation to the T lymphocytes. However, these classes differ in their tissue distribution and processing of antigenic peptides. you could see class 1 MHC pathway, peptide MHC class 1, endogenous antigen, proteasome, TAP, Golgi. Then MHC class 2 pathway, peptide MHC class 2, endogenous and exogenous. MHC class 2, endoplasmic reticulum, Golgi, exocytic route, endogenous antigen, CD8 plus DS only cross presentation, peptide MHC class 1, exogenous. Antigen processing and presentation pathways in dendritic cells, MHC1 pathway. Intrinsic pathogens like viruses or endogenous proteins are either degraded in cytosol or nucleus by immunoproteasomes and are presented as peptides along with MHC1 to CD8 plus TCTL cells. MHC1 pathway, the exogenous peptides are endocytosed and via endomembrane system binds to MHC2 to the presenter to CD4 plus T cells. Cross presentation pathway indicates that the exogenous peptide, which has been sequestrated via endocyte mechanism, may compete for the peptide either to MHC class 2 or MHC class 1 pathway. Some diseases have a unique ability to deliver exogenous antigen to the MHC class 1 pathway. Class 1 MHC antigen presentation. The endogenous antigens are first degraded by cytosolic and nuclear proteasomes. These are immunoproteasomes, which are mainly involved in extending the peptide pool rather than be selective for MHC1 peptides. This explains the wide diversity of peptides presented. The peptides thus formed are transported via transporter associated with antigen presentation. TAP, and resemble in ER with the MHC1 chaperone molecule tapacin. The antigen of the peptide to MHC1 molecule. The binding of the peptide to MHC1 molecule is aided by two or more chaperone proteins, calreticulin and ERP53. The complex is formed after loading of the peptide along with tapacin, MHC1, ERP53-7, and the calreticulin is called peptide binding complex, PLC. The MHC class 1 complex is finally released for the chaperones and is transported to plasma membrane. Class 1 pathway. In the MHC class 1 molecular pathway, endogenous proteins are broken down by proteasomes into smaller peptides. In the MHC class 1 molecule pathway, endogenous proteins are broken down by proteasomes into smaller peptides. In the endoplasmic reticulum, an antigenic peptide binds to the peptide binding site in an MHC class 1 molecule. The peptide MHC complex then migrates to the Golgi apparatus to the cell surface. Then class 2 MHC antigen presentation. MHC2 molecules are synthesized de novo and assembled in the ER. They are transported to Golgi where they associate with the chaperone in invariant gene. MHC2 Li complex move to the endomembrane system where together with other chaperones DM and DO regulate the loading of peptide within the MHC2 peptide binding cleft. MHC class 2 associated Li peptide clip remains associated with the peptide binding groove of MHC heterodimer until DM catalyzes the dissociation of clip and binds the processed antigen at a low pH. HLA-DM then transfers the clip with the specific peptide from the exogenous pathogen brought in through the endosomal pathway to be presented to CD4 plus TH cells. This cytosol to endosome transport may lead to the processing of proteins translated into the host cytosol by intracellular pathogens. In the class 2 MHC molecule pathway, Alpha and beta subunit of MHC class 2 molecule bind to the invariant gene and the LI is partially degraded in an endosomal compartment. The portion of Li which occupies the antigenic peptide bind site to the MHC class 2 molecule called CLIP is removed with the help of HLA-DM, freeing the molecule for binding process to antigen. Once antigenic peptide has bound to the MHC molecule to class 2 molecule, the complex migrates to the cell surface. Role of MHC non-self-recognition. T cells cannot distinguish between self and non-self peptides. During the early development, most of the immature undifferentiated T cells from bone marrow that encounter self-antigens are eliminated. The maturation and proliferation of T cells happen in thymus via random gene rearrangement of TCR gene. This random generation of huge repertoire of T cells also generates 95% of those cells that bind to self-antigens and are eliminated. The only T cells that mature and enter the circulation are those that recognize peptide MHC complex. Thus, through thymic selection, MHC genes influence the development of T cell repertoire. Genetic architecture of MHC. The MHC genes are located in autosomes, Y chromosomes, and even in mitochondrial genome. It is a large chromosomal region of chromosome 6 that has over 200 coding regions, and apart from specific immune functions, these genes also impact growth, development, reproduction, order, and olfaction. The MHC in mice is termed as H2 histocompatibility complex, and in humans as HLA, human leukocyte antigen complex. In immune response, specific regions of MHC gene clusters, also called a classical, are closely linked loci called class 1 and class 2 loci, and are the most divergent among invertebrates. Among the most studied, humans have 6 class 1 loci, alpha, beta, and C. The 8 class 2 loci, DP, DQ, and DR, whereas mice have 3 class 1 loci, K, D, and L, and 2 class 2 loci, A and E. The polymorphism can be observed in over 170 to 100 alleles per locus, with each locus having multiple coding genes. So close linkage between these loci means each individual inherits a particular combination of MHC alleles as a single haplotype. Thereby, there are little or no chances of combination separating this loci, resulting in highly polymorphic status. This condition suggests that MHC are inherited from a common ancestor and is being maintained by natural selection. Till date, it is extremely difficult to unravel the functional aspect of each loci, owing to high gene density, extreme polymorphism, strong linkage disequilibrium, and clustering of genes with related functions. Hence, our understanding of MHC-related diseases are very poor, because natural selection should have eliminated the neutral and deleterious alleles and must have kept only the disease protective allele. With specific variation among the array of MHC gene variation is functionally relevant. MHC gene-related diseases may not be in the gene, but the discrete changes in the amino acid or the changes in the groove resides. Natural selection and MHC genes polymorphism. There are many evidence that confirm that MHC polymorphism is maintained due to natural selection. First, high number of alleles in these gene segments or gene densities may arise when neutral alleles are not selected and are randomly fixed through genetic drift. Second, there is a lot more gene density and uniformly in the proportions of allelic frequencies. Third, the selection pressure resulted in fewer MHC homozygotes, which are not expected of a randomly mating population. Fourth, high linkage among allelic loci in individuals and their strong linkage disequilibrium are more often than chance expectations. Fifth, the high DSD and synonymous substitution ratio in the antigen binding site of MHC genes to both the neutrality and purifying selection maintains diversity. Since the diversity of MHC allele is pretty ancient in evolutionary timescale, thus the persistence of class 1 and class 2 MHC diversity is clarified for Darwinian natural selection process. MHC polymorphism though is important for disease resistance, it is also involved in promoting outbreeding. MSC polymorphism is aimed to provide herd immunity. If large number of variables are present in a population, then it is more likely that the infection may not get an escape route and the spread is restricted. Thus, it can be related to the potential of certain populations to their relationship with the pathogen associated selection. MSC polymorphism among humans can also be explained on the basis of admixture with archaic humans. Whenever a new species is territorially isolated, it acquires a new set of MHC alleles, thus involving novel alleles. The best example may be given in human evolution, where HLA-B73 allele of HLA-B found in West Asian population was being traced to Denisovans, who are related to Neanderthals. Hence, certain haplotypes of MHC were introgressed into Eurasian and Oceanian populations much before they were introduced into the African population. The polymorphism among MHC has been useful in tracing ancient population migration. For example, this can help reduce the divergence of early humans from their African origins. Epigenetics and MHC. At times, it is difficult to explain certain phenomena through conventional genetics, and there are such cases where expression of MHC gene was shown to have an epigenetic effect. The famous example being that the offspring of mothers carrying the affected allele of MHC2 HLA-DRB1 15 for multiple sclerosis and autoimmune disorder are more likely to get infected rather than fathers carrying the affected gene. The master regulator of MHC2 genes has been the MHC2 transactivator, which play the pivotal role in normal immune function. Significant epigenetic modifications such as methylation, acetylation, and histone modification have been demonstrated to alter C2TA expression. Therefore, many diseases associated with aging and autoimmunity is linked to polymorphism of C2. MHC in transplantation. Allogenic immune response. When a tissue transplant is performed, the HLA of the donor is recognized by the recipient's immune system, triggering an allogenic immune response. The divergent alloantigens caused due to large numbers of genetic differences between donor and recipient are the easy targets for T cell recognition, thereby increasing the risk for both graft rejection and graft versus host disease (GVHD). Allogenic immune response may happen by three recognizing mechanisms. Indirect recognition. The donor's HLA is processed by APC and presented as peptides with HLA molecules to be recipient's T cells. This mechanism is mainly dominant in chronic rejection. Direct recognition of donor's HLA molecule is directly recognized on the donor's presenting cells. Receptor recognizes the foreign HLA as own molecule with foreign peptide. This causes acute rejection. Semi-direct recognition. The immunoglobulin-like receptors as NK cells recognize the polymorphic sequence of HLA-AC, B, or A in target cells, causing acute rejection. So it has been shown recently that the naive and memory CD4 plus and CD8 plus T cells are cross-reactive against allogenic HLA molecule presenting self-peptide. In HLA testing and clinics. Additionally, HLA typing was performed by serologic testing by using anti-serum and complement-dependent cytotoxic assays. Lately, more precise DNA-based HLA typing methods using molecular techniques have been developed. Techniques like sequence-specific oligonucleotide probe hybridization, sequence-specific primer amplification, sequence-based typing, and reference strand-based conformation analysis are frequently used. The high polymorphism among HLA with more than 800 alleles have restricted to standard HLA typing at the HLA-A, HLA-B, HLA-C, HLA-DRB1, and HLA-DQB1 genetic loci. An HSCT donor is referred to as 10 by 10 allele match or perfect match, and both HLA alleles are identical at each of the HLA-A, HLA-B, HLA-C, and HLA-DRB1 and HLA-DQB1 loci. While searching for an unrelated donor, high resolution 4-digit genetic typing of both the patient and the donor is necessary. Now let's have a summary. Major histocompatibility complex or MHC class 1 and 2 loci are the cell surface receptors encoded by the most diverse gene loci in vertebrates and spans about 0.1% of total genome containing 200 coding loci. The MHC gene are located in autosomes, Y chromosomes, or even in mitochondrial genome. It is a large chromosomal region of chromosome 6 that has over 200 coding regions. The MHC in mice is termed as Histocompatibility complex, and in humans as HLA, human leukocyte antigens complex. MHC not only distinguishes self from non-self but also plays a pivotal role in tissue transplantation and rejection, autoimmunity, and susceptibility to infections. Apart from specific immune functions, these genes also compact growth, development, reproduction, order, and olfaction. The MHC class 1A genes are encoded by three genes HLA A, B, C, and present small peptides to CTLs, cytotoxic T lymphocytes, enabling each cell of the body to the immune system with possible chances of infection. Therefore, every nucleated cells of the body present MHC1 molecule. MHC class 2 molecule peptides are expressed on specialized cells called antigen-presenting cells APCs, mainly the macrophages, dendritic cells, and B lymphocytes. These molecules are encoded by three polymorphic genes HLA-DR, HLA-DQ, and HLA-DP that bind to different peptides. Both MHC1 and MHC2 present peptides to the CD8 plus and CD4 plus T cells. The sources of these peptides may differ. The MHC1 are presented with intracellular peptides and the exogenous peptides are presented by MHC2. Both MHC classes have similar three-dimensional structure. They originate from the common parent gene by gene duplication and a similar function in antigen presentation to the T lymphocytes. MHC genes influence the development of T cell repertoire through thymic selection, as only those T cells that recognize peptide MHC complex are able to mature and enter the circulation. There are many evidence that confirm that MHC polymorphism is maintained due to natural selection, like high number of alleles, uniform allelic frequencies, deficiencies of homozygotes, linkage disequilibrium between loci, high DNDS distribution, ancient allelic linkages, and association meeting preferences. So, expression of MHC genes was shown to have an epigenetic effect, as many diseases associated with aging and autoimmunity is linked to polymorphism of MHC2 transactivated. Allogenic immune response happen due to divergent MHC alloantigens between donors and recipients, which are the easy targets for T cell recognition, thereby increasing the risk of both graft rejection and graft versus host disease. GVHD, precise DNA based HLA typing methods using molecular techniques like sequence-specific oligonucleotide probe hybridization, sequence-specific primer amplification, sequence-based typing, and reference strand-based conformation analysis are frequently used for HLA typing in clinics.